Thank you for this very kind introduction. Let me thank you for giving me the opportunity to speak about morphological process control as, I would say, an interesting possibility, an interesting perspective in the life sciences. So I somehow, yeah, okay. You all have seen pictures like this, man. This is conventional engineering. As a matter of fact, in conventional engineering, you always try to via sensory input to produce some sort of representation of your robot or whatever system you're interested in. And then based on this representation, you plan what's going on. And finally, you actuate the system. And probably the system should do what we would like it to do. In order to achieve this, the goal of conventional robotics is always to reduce or at least control the physical dynamics. It should be as only very few amounts of what we usually call noise. And as a matter of fact, this usually leads to robots which are heavy and stiff. You in morphological control do it the other way around. You turn the enemy into the friend. That means you exploit physical dynamics. And as a matter of fact, this has the interesting consequence that also soft structures are welcome. You all know pictures like this, and I think it's fair to say that morphology then mostly accounts to a combination of classical mechanics, shape, plus material properties, plus other aspects of your system. And the question is now, can we extend this notion of morphological control, I would say this paradigm of morphological control and computing, into the life sciences? And I think, yes, we can, but morphology has to be understood in a different way. As a matter of fact, on a cellular or at least on a microscopic level, morphology is the combination of chemistry, spatial organization, self-assembly, supramolecular compounds, membranes, and lots of other things. And the importance of spatial organization can, for example, be seen if you analyze how a eukaryotic cell is working. When you see here a picture that I have stolen from Encyclopedia Britannica, in which you see a eukaryotic cell, which is quite well organized and contains a lot of individual compartments. Now, we have had a large European project, ACDC project, artificial cells with distributed cores to decipher protein functions. This project has already finished. And the basic idea was to set up compartmentalized microscopic reaction environments. Our inspiration came from the cell, such as, for example, from organelles, as you can here see on the left-hand side. This is a Golgi apparatus. That's a very important device, mechanism or compartmentalized structure in the cell, which helps us to build up branched oligosaccharides in our body. And, well, we wanted to build up a structure which could do, well, probably not exactly that, but something that at least resembles structures like this. Needless to say that this is maybe still slightly a naive picture of a cell, just to think of it as a combination of microscopic reaction environments which are filled with fluids. Maybe it's better to think of a cell as sort of a highly organized gel, but for process management, cellular process management, it may be sufficient to think of compartments filled with fluids. In this project, ACDC, we have developed two different approaches to that. One of these approaches, I call it the Trento foam. People in Trento hate this. They would like to call it the Trento system, but actually it consists of vesicles. Vesicles are liquid volumes, basically liquid volumes, which are surrounded by a lipid bilayer, and this lipid bilayer can be decorated with linker sites so that you can have the selective linking between different vesicles, and you can also equip them with pores so that you can get material transport between the different compartments. Another system, well, I have to say that this is sort of a stochastic compound. The linker sites, they are selective and they lead to sort of a structured environment. In fact, well, you have a correlation structure, but you have not really, let's say, geometric arrangement. That's completely different here in our Cardiff system that has been developed by the University of Cardiff. They use small droplets which they immerse into a fatty environment. And if you do that, then you get a lipid bilayer between these two droplets. And this lipid bilayer can be equipped with pores. And using microfluidics, you can set up in a reproducible manner geometric structures which well-defined which a well-defined connection structure and which form a well-defined network. Now, presently, we extend this work to a project that is called Bio-HhOST. It's a next generation 3D tissue models, biohybrid or hierarchical organoid synthetic tissues comprised of live and artificial cells. You realize I have to still get acquainted to this new acronym because the project just started this February. And the basic idea is, well, we want to build up a platform for the analysis of the behavior of biological cells. So, for example, tumor cells. And if you do that, many always have the problem. You can study tumor cells either isolated, but unfortunately, isolated cells, they behave quite differently from cells that are embedded into a tissue environment. Well, you could study tumor cells in a tissue environment, but that is for another reason not such a very good idea because it's very difficult to standardize tissues, and that means that the interpretability of your experiments is somewhat reduced. And so the idea of Bio-HhOST is we use our well reaction compartments that we have built up in ACDC, and we build something that resembles a tissue-like, that is sort of a tissue-like environment, a tissue-like environment which looks to the biological cells as if it were a tissue. Let's hope that the tumor cell behaves as it were embedded into a real tissue. But of course, because we produce these compartments in the laboratory, we can do that in a standardized and reproducible manner. Now, that's all well and fine, but here we speak about morphological control, and I think it's fair to say that we have to think a little bit deeper about how can we actually exert control by well, using sort of chemical microcompartments. And the first idea is pretty simple. Assume that you have one, that you have just one reaction compartment and you have a complicated reaction network in this one compartment. It's clear that you have to fit reaction conditions or chemical environment conditions, which are one fits for all conditions, probably for the individual branches of this reaction network. You will have suboptimal conditions. And obviously, what you can do is you can split up your compartment into many different compartments such that the individual steps of the reaction can take place under optimal or otherwise, let's say, controllable structure so that you can exert a certain amount of control over your reaction process. Of course, nothing is for free there, besides the fact that you can optimize your conditions or at least control your conditions. Yeah, there is a con. You have to organize the transport of matter and information between the different compartments. And that's, of course, quite a thing. Assume for a moment that you can do that. Well, then already, if you have this possibility to have different reaction compartments or different types of reaction compartments, you can exert two types of control. First, you can choose the types of your compartments. And second, if you assume that there is only matter transport between adjacent compartments, then of course, just by the arrangement, the geometrical arrangement of your compartments, you can get a certain control over the sequences of reactions that you perform. And that gives you control just by patterning. Now you could ask, well, why don't you do that in a test tube? Would be much easier. Well, as a matter of fact, you see, assume, for example, let's take catalysts as an example. Well, lots of molecules in the laboratory, they have to fulfill at least two requirements. Catalysts have to fulfill two requirements. They have to be effective and they have to be stable. If you have a catalyst, if you produce a catalyst which has a half-life time of, let's say, 120 seconds, then this catalyst is of no value at all in an industrial or in a laboratory setting, or it becomes very expensive to exploit it. But 120 seconds, that's quite a bit of time on the microscopic level, even if you only rely on diffusive transport. So that means on a microscopic level, the range of molecules which are interesting is much, much wider because stability is not anymore that of an issue. Now, how can we study control? Always doing experiments is very difficult because experiments are terribly complicated. And therefore, I think you from the artificial life scene, it's probably not too difficult to advertise this here, but of course we should do artificial chemistry. Artificial chemistry can help us to identify relevant dynamics and interesting system states and relevant parameters in a system. And of course, we can exploit the structural similarity between different reaction systems. And here you see one of the big champions of the field, Wolfgang Banzhaf, whose work I highly admire. Now, what is this good for and what is this to do with this ACDC project? Well, let's take for an example the, I think, quite well-known Kaufman system. What has Stuart Kaufman found? Well, Stuart Kaufman, he analyzed polymers that are composed from monomers, which are called either A or B. And depending on their sequence, these monomers can have sequence-specific catalytic abilities, cleavage or ligation of other A-B sequences. And then Stu did a sort of a graph theoretical analysis in a somewhat seminal paper in this field. And well, he found that the longer the sequences are, the higher is the probability that you see an autocatalytic reaction network. Problem is, in the laboratory, you see none of them. So the question is, where are all these systems? And here I have to say in advance, Stuart Kaufman knew everything in advance. Before I did the work, he was certainly aware of that, but nevertheless, I came then up with the idea, well, maybe a static analysis of graphs is probably not the best thing if you want to see the probability for the emergence of an autocatalytic network. Maybe you should also take into account the question whether a reaction system is stable, and that's a dynamical issue. And then I started to analyze the dynamics of these systems, and then I came up with the conclusion this was something Stuart Kaufman did not yet know, that whether or not you have a high probability for an autocatalytic network to emerge depends critically on the kinetic reaction rates, or, which is more or less the same, the probabilities for being catalytically active. And particularly kinetic reaction rates are difficult to influence. But what you can do if you're in a spatially heterogeneous system, what you actually care about are effective reaction rates. And effective reaction rates are the result first of the chemical kinetic reaction rates, and of course the diffusion rates, because the effective reactions are a product of chemical reactions and transport processes. And these transport processes, they are relatively easy to influence in an ACDC-like spatially structured environment. And then we used artificial chemistry to study such systems in a compartmentalized environment, and we have seen that by fine-tuning reaction rates, we easily can turn non-functioning systems into stable ones. Unfortunately, there are not yet experiments that corroborate our clues. For evolutionary purposes, for stabilizing purposes, spatial structuring is really important. A couple of years ago, we analyzed the stabilization against molecular parasitism, stabilization of genetic codes. That means genetically translation and replication systems. And we have seen that under suitable conditions, systems can be stabilized, which otherwise are not stable. And that is relevant for studies of the origin of life. But of course, we are not only interested in the origin of life. As a matter of fact, I think one of the goals that we have in technology is to achieve production technology for what I would like to call multi-scale matter. As a matter of fact, most matter that we have today in technology is somewhat built up from molecules or atoms and has somehow no real hierarchical structure, whereas most lifelike matter has a hierarchical structure. And I think that, well, a bio-inspired, bio-like production process is probably the only way with which we can produce materials that resemble the structure of wood or other very pretty things. And there are a lot of advantages of lifelike matter. I have summarized here on one side a number of them. I really want to highlight that, let's say, all the different alloys that we use, they somehow rely on atomic properties. So we have to use lots of different elements, lots of different alloys, whereas lifelike matter basically consists of carbon and nitrogen and hydrogen and all these very abundant substances. And, nevertheless, life has pretty amazing properties, as we all know. Now, I spoke about self-assembly, and I think that self-assembly is one of the most underestimated fundamental processes with respect to also information processes. A couple of years ago, I wondered, why is it the case that children can multiply more or less arbitrarily large numbers? As a matter of fact, if you compare children to evolving digital circuitry, then you realize that to evolve digital multipliers is comparably easy as long as the numbers you want to multiply are comparably small. The longer these numbers become, the more difficult this evolution process becomes. And as a matter of fact, it becomes exponentially difficult. And the situation with children is completely different. Man, children have quite a hard time to learn basic multiplication table and even a harder time to learn the algorithm with paper and pencil. But once they master this algorithm, they can multiply more or less arbitrarily large numbers. And then I wondered, well, why is this the case? Then I realized I spoke to an electrical engineer and he drew me, well, the way how multipliers are built with digital circuitry. And then I realized, oh yeah, that's a pattern actually. That's a pattern of fundamental, very simple logical functionalities. And then I realized that actually what here is done is you transfer the global logical structure of a problem into the geometry of the arrangement of function generators. You turn logic into patterns. And then I asked myself, well, is it easy to evolve patterns? And as a matter of fact, it is. We came up with work in which we studied so-called self-assembling logical blocks. We did that in silico. These logical blocks are equipped with recognition sites and function generators. And well, we could evolve scalable multipliers, but also lots of other types of functionalities. And the interesting aspect here is that in order to do that, we had to develop a specific evolutionary algorithm, an evolutionary algorithm that somehow promotes the learning of structures over the learning of individual instances. Hello, whoever that was. So now let me turn to something I'm particularly interested in nowadays, and that's the communication between us scientists and the rest of society. Already Sabine came when she spoke about the ethical aspects of our science, and I think this is really of high importance, the ethical aspects and particularly also the communication. I'm very proud of this slide. Here you see that's a report from the Bundesrat. What is the Bundesrat? Well, that's our government. Our government, we don't have a prime minister. We just have 7 ministers and they form the highest board of Switzerland, so to speak. And during the pandemics, I was quite strongly involved in the simulation of these pandemics. And here you see the concept paper of the Bundesrat for the relief of the lockdown measures, the 3-phase model. And well, the calculations therein, the modeling therein, that I was part of the team that did this modeling, and actually my wife has chosen these colors in the graphs, and the Minister of Public Health of Switzerland really liked these colors. These were my 15 seconds of fame. Now, by the way, uh, all our calculations have been strongly supported by Public Health England. That's the reason why we were so good, because we had the numbers from Public Health England. And unfortunately, this is, by the way, the truth. Yeah, but what did I learn during this period? I learned something that is absolutely crucial in whatever type of modeling you're doing. The first and most important lessons for me with respect to scientific collaborations is that variables are not observables. If you do abstract modeling, if you do computational modeling, you have to be aware of the fact that you choose model variables, and these model variables have to be brought into context with actual observables. And you have to you think about this connection right from the start. Most modelers don't do that. And this is one of the reasons why modeling quite often fails. Bringing together model variables and observables is probably not that difficult in, say, mechanical engineering, because then you speak about position and velocities and stuff like this, which is easy to measure. But as soon as you go to the life sciences, this problem becomes much more difficult, because in your model, you probably have chemical densities, something like that. In your experiments, you get fluorescence images, and you have to think of how to bring fluorescence images together with your model variables. And if you set up the model, you have to think about that right from the start. A second lesson, which is important when you speak to the public, to us, I think this is clear. Modeling is more than forecasting. With a model, sometimes you can predict the future, but there are many more reasons why you should model. You can optimize the present, you can explain what you see, you can analyze the dynamics, you can produce data for modern controllers, you can detect emergent structures, and so on and so forth. It's important to explain people what models are good for. Also important, decision support is not a scientific collaboration. We, as scientists, we have to learn that if we communicate with the public, or let's say with different spheres of society, here I follow Luhmann's interpretation of different spheres in society. If the sphere of science communicates with the sphere of, let's say, politics, we have to be aware of the fact that these people there, they are not dumb because they don't speak science English. We have to find a way of collaboration. Very important, especially if we collaborate with politics. We have to give options, not advice. See, there are value statements and factual statements. And we as scientists, we are responsible for the factual statements, the value statements. At least in a democracy, we have institutions that are responsible, time partly even elected. They have to do the valuing. We have to do the facts. In that respect, it's very important to explain that defining optimality is something quite difficult, sometimes quite difficult. Politicians always ask us, well, give us the optimal solution to a problem. Well, then you have to ask, well, what actually do you want to optimize? And again, politics has to answer the value statement, what exactly they understand by optimality. We have to say how to reach optimality based on the facts we know. Now I'm close to the end. And I think it's very important, if you do a model, we have to be aware of the fact that our model always tells a story. What I want to say here, for example, in epidemiology, if your model is based on data that is subdivided according age, well, then your model tells a story about age. Your model always tells a story. If you use a model that is based on data that is subdivided according to age, for example, your model will tell a story about age. This model, this story may differ from a story that a model tells if it uses data that is subdivided according to, say, for example, socioeconomic status. At the end, we have to make it clear when we speak to the public, particularly to the media, that also the readers do have a responsibility. And I think we should try to build up an ethics of interpretation. Finally, morphological control. I come to my conclusion. Morphological control is more than steering robots. Actually, it can be applied in the life sciences. We can learn something for the life sciences. Interesting chemistry happens in spatially organized systems. That's what biology tells us, and we try to mimic it with the use of small microscopic compartments. Self-assembly, I think it's underestimated. You transfer, it's a transfer of functions into patterns. Lifelike matter, that's, I think, a way we should go. And with this, I would like to thank you for your attention.